Hello, everyone. I am Deborah S. Capobianco, and today we will be discussing the Nobel Prize associated with the circadian rhythm. The 2017 Nobel Prize in Physiology or Medicine was jointly awarded to Jeffrey C. Hall, Michael Rosbash, and Michael W. Young, and this award was for their discoveries of molecular mechanisms controlling the circadian rhythm. Well, the circadian rhythm is the body's clock that helps in regulating physiological functions throughout the day. And what does that mean? Well, physiological functions can refer to anything in your body that helps you function throughout the day, such as the rise or fall in temperature, how focused or alert you are, the blood pressure. There are so many things that go on. The list can go on and on. But the biological clock, this biological clock, has a connection with how we function and how well we function or how poorly we function as human beings. Is they were able to find the minuscule molecular reasoning behind how this clock functions. So in a 24-hour day, we function based on time. So we wake up at a certain time, you go to school at a certain time, you do homework, you eat, you drink. All of these activities have an effect on your body, how it functions, and again, how well you are focused and how alert you are. The location of the clock is within the body in the cell nucleus. How did we get all this information about the circadian rhythm, this cool physiological function controlling biological clock? Well, the Nobel laureates did have some help. We have to go back a little bit in history for this. Jean-Jacques Dortu de Marin in the 18th century, he worked with this plant, the mimosa plant, and found something interesting. What he did was he left the plant out in sunlight during the day, and at night he left it there, but it was dark outside. So during the day the leaves would be open, and during the nighttime he noticed the leaves would close of this mimosa plant. However, interestingly enough, when he moved the plant to complete darkness, constant darkness throughout the day into the night, the rhythm still continued. What does that mean? Well, the leaves stayed open during the day, still, and then at night they closed again, even though it was still constant darkness. So this tells us that maybe external conditions don't always affect this clock. Perhaps it's more internal. In the 70s, Seymour Benzer and Ronald Konopka, they discovered a gene called the period gene by studying fruit flies. Now, these fruit flies had mutations in their period genes that would interfere with their circadian rhythm. So their sleep and wake cycles were off. So they would sleep at different times and wake up at different times on different days. It wasn't always the same, and that's purely because of this mutation in their period gene. We can also, at this point, start to figure out why the circadian rhythm got its name. So circa, part of circadian, means around, and dies means day. And just as a review, mutation is a change that occurs in our DNA sequence due to mistakes when it's copied, when it's processed. You can think of it as a recipe that has gone wrong. Maybe different ingredients were added or they were switched up. Instead of baking soda, maybe you used baking powder. Instead of dark chocolate, maybe you used white chocolate, and something completely different happens. But with DNA mutations, it actually causes dysfunctionality, and that's what this specific period gene mutation was causing in the fruit flies that were being studied. They figured out the internal functionalities of this biological clock. So how did they do that? Well, they definitely used history to their advantage. After looking at the information from the 18th century and the 70s, they isolated the period gene and called the protein that it makes PER. An easy way to remember that is the first three letters of the word period. Now, the PER protein, they realized, was the main protein that was responsible for this circadian rhythm. And there was also a pattern of the levels in this PER protein. During the day, the levels of PER were usually lower than during the nighttime. Anything that is cyclic in nature, or anything that is a cycle, if you think of the life cycle or think of the water cycle, where you have rain, which evaporates, goes back up, it's a repetitive process, but there's always something that starts it and stops it, something that keeps it going, something that maybe slows it down. You can think of how those cycles function in order to try and understand how the circadian rhythm functions as a cycle. The molecular components starts with the help of light. As long as we have that, just remember that that's how the activation actually starts. Now, if you think of this as a recipe also, that might also help. So you have step 1, where the PER protein is regulated in this cyclic manner that we were talking about through inhibitory or negative feedback. You're going to have its mRNA. It's just synthesized, and when something is synthesized, it's made. So it's being made, it's being processed. mRNA is made and it enters the cell. Now, what does that mean? Well, basically, once the period gene is activated, the period gene's genetic material is entering the cell. So our cells, our human, animal, or plant cells, that's how it enters the cell. And then that's where the PER protein is generated. Next up, we have a protein called DBT protein, or double time protein, controls the amount of PER that is being generated. And then the TIM protein, or timeless protein, comes in, binds to the PER protein, and it allows it to enter the nucleus of the cell. And then finally, period gene activity inside the nucleus. Remember the mRNA that we talked about? The activity that mRNA of the period gene that was being created is halted. So it's kind of a pause, because remember, this is a cycle. So it's not a stop as in it completely stops and never happens again. It stops for the time being until light is introduced again, and then you start the whole circadian rhythm, or the clock, the cycle, again, all over again. So this whole cycle, put things in perspective, is similar to kind of having a thermostat, and it's going to come back to, remember we talked about inhibitory feedback, the negative feedback, similar to having a thermostat in your house. When you set a temperature, and once the temperature in the house reaches that set temperature, for example, 70 degrees, let's say, it turns off. Now, that's something electrical that's happening, but if you think about it, since our body is already set for certain things, this is what's happening on a molecular level. It's pretty cool. Now, inhibitory, the word inhibit means to hinder or restrain, prevent an action or process. In this case, we're hindering or preventing too much buildup of PER protein. Because remember, if we have too much PER protein, the cycle won't last as long, and we would also fall asleep sooner. Maybe that was something that was happening in the fruit flies from that genetic mutation. And then we have the word feedback. You may have already heard of that word. It's just a reaction or a response to something. In this case, a particular activity, and it's negative because it opposes the process. So in this case, the feedback is negative because it is going against the buildup of PER instead of accelerating it. Instead of making too much, it's trying to slow it down. So that's why it's negative feedback. So I hope that helps in understanding how this whole process works, and how inhibitory feedback is a very, very important thing over here. Now, PER is regulated in a cyclical manner. Just like we talked about before, it's a cycle through negative feedback. And we come back to the thermostat analogy because it's kind of responsible for turning itself on and off. Now, we have to be careful because we did mention the DBT protein and the TIM protein. So it's not that the PER protein turns itself on and off by itself, but it does pretty much, that's the main protein that is required for this cycle. it has help. So maybe you can think of the thermostat having help of other software that's built in order to turn on and off based on the temperature it's set for. So these sleep or wake cycles completely controlled by this inhibitory feedback. You need that negative feedback. If not, it's going to completely throw off the 24-hour cycle that we have, and therefore throwing off the circadian rhythm. And this is also a good place to try and understand why jet lag plays such a huge role when we get tired from vacationing in a different time zone, or even just a one-day travel in a different time zone. And we're going to bring this all together and look at the big picture. So the big picture, starting from the beginning, from me trying to see if you were focused enough to see the alarm clock on the top right corner, to learning about the whole process of how the circadian rhythm works and what the biological clock is, even for each and every one of us. If you look at it, there are a few factors to consider that do affect the functionality of this clock. Now we know how the molecular activities, what they do, but there are things that we can do that help also control or schedule these molecular activities. So if you sleep on time, depending on how much light you are around when you wake up, how much darkness there is, so that when you go to sleep, you have a nice sound sleep. You can tell you have this whole section where it says deep sleep. Now, during that deep sleep time period, it will be great to have total darkness so that you have a very nice do not disturb type of sleep. And then we look at having a possible schedule, even if you're not used to waking up at a specific time every single day. It might help to set your alarm and at least try to teach your body to be aware of your surroundings at that time. Even if you hit the snooze button, it might still help. Just because there are so many other factors at play, like blood pressure and cortisol release and temperature. These are things that you don't necessarily want to fall sick for or throw off just because you don't want to control what time you wake up or go to sleep or how well you sleep. And then last but not least, the last two are actually coordination and awareness. If you do your best to stay coordinated with your body and you're aware and you listen to your body, that is going to be the main thing that will help you get used to this circadian rhythm and figure out how you function to increase focus. These are the references that I used for the pictures and the Nobel Prize article. Please go ahead and go through. And thank you for your time today. I hope you enjoyed learning about this as much as I enjoyed learning about it as well.